Hello, everyone. We are now live. I'm just going to wait a few minutes before we actually start the seminar. All right. And it's noon. Hello, everyone. Welcome to this new LIMNO seminar. Today we are joined by Gwyneth MacMillan. Gwyneth is a PhD student at the Université de Montréal in ecotoxicology of mercury and rare earth elements. Gwyneth did her undergrad at the University of Western Ontario in biology and anthropology. She's been doing some very exciting work up in the Arctic, and that's what she's going to talk to us about today. Gwyneth.
Okay, let me just share my screen with you. Let's see. Is this working? Can you see my slides?
I can see your slides very well.
Okay, excellent. So thanks for being here today. I'm really looking forward to talking to you about rare earth elements. Initially, I was going to talk about mercury, but this is really exciting data. We're actually just going to submit the paper tomorrow, so it's really hot off the presses. So hopefully you'll find it really interesting. And it's pretty new, so I'll take you through some of our main results that we've been finding. The talk is called Rare Earth Elements in Marine, Terrestrial, and some Freshwater Subarctic Ecosystems. And I'm a PhD candidate, as Alex said, at the University of Montreal. You can contact me here by my email or on Twitter as well. And just let me know if the slides are changing fine too. I work co-supervised in my PhD by Marc Amiot, who's at the University of Montreal as well, but also for the Center for Northern Studies. And I also have, my supervisor is John Cheddawa, who works at Environment and Climate Change Canada in Ottawa. And we also have two northern collaborators. So Joel Heath, who works for the Arctic Iota Society, and also Raymond McPegak, who's in the Kudryavik. He works for the Sakuk Land Holding Corporation as well. So I'm just going to take you through a little bit of an introduction here. So what are rare earth elements? Just thought I'd introduce them to you. They're not known by everyone as well. So the rare earth elements include the 15 lanthanide metals, as well as yttrium and scandium. And there's sort of this column of odd floating elements down at the bottom here of the periodic table. It's this first line here. And they're kind of grouped together into a category we call the rare earth elements because they have a lot of chemical similarities. They're all trivalent typically, except for a couple that have different redox states as well. And they're quite electropositive and insoluble compounds that are lithophilic, so really attached to soil and sediment. Just for the purposes of showing you my results later, I'll mention that we often divide rare earth elements into two groups. So we'll have the light elements, which are from lanthanum to gadolinium here in yellow. And then we'll have, based on atomic mass, the heavier elements. Here and there, all fairly similar elements, but there are some subtle differences between the light and the heavy elements, mainly that the light ones are more soluble and the heavy ones are less soluble. So rare earth elements have not received that much interest in terms of their ecotoxicology, in terms of their role as a pollutant in the environment. And if you compare them to other trace metals like cadmium, mercury, or lead, there's really not a lot of literature on how these elements behave in the environment. If you look at this figure, it's basically just showing year on the x-axis and the increasing number of papers published that talk about rare earth elements. So it is becoming more trendy. There are more and more articles being published on these elements, but there's still kind of a lack of basic data. Like, we don't really know that much about their uptake or the toxicity, bioaccumulation, and that kind of thing too. So essentially, this lack of data means that we don't have toxicity guidelines in Canada, and there aren't any in the US or Australia or in any major country either. So you don't really know enough about them to know how and when they'll become toxic. And why are we interested in them as well is because they're kind of hidden in all modern technology. We don't hear a lot about them, but we find them really in everything from the computer and iPhone that you're using to plasma TVs. They're used as fertilizers in agriculture and a lot of different appliances. So they're really used all over in most modern technology and medicine as well. And one really interesting use is that they're really critical for our current transition to green energy. So they're really important for magnets in wind turbines and car batteries as well, electric car batteries. So that's a really important use of rare earth elements. And in terms of their use, they're increasingly used all around the world. And this is sort of a figure showing you their potential scenarios in the future. So demand scenarios based on different historic trends, historic growth, or expert advice, or different uses of, we have aggressively used green technology or a more moderate technology. Basically, what I want you to notice is that there's a generally increasing trend for the use of these elements over time and into the future as well. And they are emerging contaminants. One of the main sources of their release into the environment is due to use in our high-tech industry, but also due to different mining and processing and basically the mining and processing of rare earth elements. And this picture here is one of the only mines that's been used for rare earth element mining in North America. It's in California, though I think it's closed currently, perhaps predicted to open in the future. And in places where we do get a lot of rare earth mines, such as China, for example, we've been able to notice increasing rare earth element concentrations in soils and water and biota all around these mines and factories. And some rivers that are near these mining areas have 3 orders of magnitude higher concentrations of rare earth elements compared to unperturbed rivers. And this is a photo from sort of the epicenter of where we get most of our rare earth ore at the moment. It's in China and Mongolia. And this is an artificial lake that's really just chock full of pollutants that sort of, I should mention that rare earth mining also produces a lot of other pollutants that are dangerous, a lot of radioactive waste, and a lot of really acidic wastewater comes out of the mining process as well. And why, so in Canada, why are we interested in understanding more about rare earth elements if most of the mining happens in China? And actually, it's true that currently China controls, over 97% of rare earth element production, but that is gives them a lot of control over prices in the market, and so there's a lot of interest in opening up potential mines all across the world in other countries, and specifically a lot in North America. And there's about 200 potential mining projects that are proposed in Canada, and many of them are actually in the north, so in the subarctic or Arctic zones. There's a huge potential to see an explosion in mining activity. And one thing to note too is that these systems in the subarctic and Arctic are already considered vulnerable. They are undergoing rapid climate environmental change. And they also are really close to potential sites for the exploration of rare earth elements. And I thought I'd show you here, it is sort of a hot topic right now. This really issue is kind of on the radar, and so one mine is proposed to go in at this place I've highlighted in northern Quebec, so about 3,000 kilometers north of where I am right now in Montreal. And it's called the Strange Lake Mine. It's by Quest Rare Minerals, and in fact, a local community that lives on the George River near this proposed mining site approached a group of researchers in Quebec, and they're wanting to set up a water quality monitoring program because they were worried about the effects on their water quality of this future mine. And so this is a project that I'll be participating in terms to see the potential effects of this mine going in and to monitor the local environment, the river and the terrestrial environment around this community. And you can see that this mine is proposing that they'll enable green technologies through a reliable supply of rare earth metals. And they're even proposing to fly their rare earth ore out using blimps because there's no roads up there. And so they have kind of a really particular niche field, and it's definitely proposed to go in the next few years up north. So I'm just going to summarize now a few of our research goals here in the study. This is really one of the first field-based studies on the behavior of rare earth elements in northern ecosystems. There are only a handful of other studies that really look at these natural concentrations. And the real research question we had was, we wanted to know, how do rare earth elements behave in a pristine northern environment? And so we wanted to measure baseline concentrations. We wanted to know how much are there really out there. We wanted to also look at bioaccumulation patterns in biota that were important to northerners. So we specifically looked at organisms that are hunted or gathered or fished by the people that live in this community that we sampled in. And then we wanted to trace the potential trophic transfer of REEs using stable isotope ratios. So these are the three things I'm going to talk about today. And this really, we are hoping, will really allow us to better understand the behavior of REEs and better evaluate the potential impacts of rare earth mining in the north of Canada. So here's a map showing our sample sites. So I'm here in Montreal on the St. Lawrence River, and then about 3,000 kilometers north on the eastern shore of Hudson's Bay. This is the community of Kuujjuarapik-Wapimagustri. So it's a community that's half Cree and half Inuit. And it's a place where the local people do still depend on traditional food sources, so food that is hunted and fished. And our sampling took place from about 2012 to about 2015. And we sampled here in this, just basically around this community. What's interesting about this project too, it wasn't only a scientific monitoring program. We actually, in 2012, we went up and sampled a series of lakes. But then after that, we set up a community-based monitoring program, and that was basically through the Northern Contaminants Program, which is through the Canadian federal government. We also collaborated with the Land Hoarding Corporation in the community and the Arctic Iota Society, which you can check out here, the website. It's really interesting. And so what we did was, it was initially the sample for other metals, and we kind of opportunistically sampled for rare earth elements as well. And the local hunters went out and they used their traditional knowledge to help design the study, to help coordinate the local sampling effort as well, and to they really had the knowledge of when and where and what species we were wanting to hunt. And so this project really could not have taken place without the collaboration of the local community in this town. And here at this picture, you can see them hunting for the common eider, which is a species that we sampled in the study. Just going to take you quickly through all of the samples that we analyzed for this study. We looked at 3 different ecosystems, because Kuujjuarapik is really a place where there's the meeting of many different ecosystems. There's a river, there's the ocean, and then there's a huge terrestrial environment with lots of freshwater lakes around as well. And so in the marine environment, we sampled marine invertebrates, so blue mussels and sea urchins as well. We also sampled the common eider and the ring seal. So those were the 4 species that we sampled from the marine environment. In the terrestrial system, we looked at both vascular plants and lichens and moss. So the vascular plants included Labrador tea, crowberry, and bearberry, 3 species that are commonly collected by the local community. And then we looked at 3 terrestrial vertebrates, so snowshoe hare, willow ptarmigan, which is similar to a grouse or a pheasant, and a caribou up north. And finally, for the limnologists, we looked at freshwater ecosystems. So we studied 8 lakes, and we had bulk zooplankton samples as well as a mix of benthic invertebrates from these systems. We also looked at brook trout. We had them in about 5 of the 8 lakes, and we had some fish that came from the river. And so these fish would spend some time feeding in the marine environment and some time living and feeding in the freshwater river as well. So I'm going to move on to the results. This whole talk should be about half an hour, so I'll be willing to answer any questions then. So when we looked at these results, the first question we really wanted to ask is, do these elements behave as a coherent group together? So how do the 15 elements behave? Are they individually behaving differently, or do they behave sort of as a coherent group? And why we're asking this is because typically the rare earth elements are known to follow what people call the rare earth pattern or the sawtooth pattern. And this is like a highly conserved abundance of all the elements are often found together in ore deposits, and their relative abundances will be very similar. And what typically you see is strongly conserved in most abiotic matrices, so in water, soil, and sediments. You'll see, so here I've plotted the 15 elements here on the x-axis and the concentration, log concentration of rare earth elements here on the y-axis. And you can see that what we typically see is this decrease, log linear decrease in concentration with atomic number. And they're all strongly they co-vary together very strongly. So the abundances are all very similar. And this is something that we can see in the literature is found in marine, freshwater, sediments, and soils, but there's not a lot of data to see if this also occurs within organisms. So when rare earth elements are bioaccumulated, do they also follow this same pattern? Oh, and one thing we can do that's often done actually is to normalize the data. So it's often hard to interpret these sawtooth graphs. So if you divide all the concentrations by a standard rock or shale concentration, you'll get a straight line. And basically what you're looking for is any deviation below or above the straight line will tell you that one element is not behaving like the other, so that there's an enrichment or depletion in a specific element. And this is our data for the lakes. So this is these 8 lakes that we sampled, and you can see that there really is no deviation from a straight line. All the elements behave together. And then when we looked at the organisms from these lakes too, we found the same pattern. So this was really interesting to see that when rare earth elements will be bioaccumulated into organisms, we see the same sawtooth pattern, and when we normalize, we see some variation, but also not any major deviation from a straight line here. So this indicates really that they co-vary together. They're behaving in a similar way, the 15 elements. And just to confirm this here, what I want you to notice is that we plotted the concentration of the light elements here on the x-axis, and on the y-axis, the heavy elements. And this is showing the same thing. So we really notice that the rare earth elements behave as a group. Their relative abundances are related to each other. So when we have increasing light concentrations, we also have increasing heavy concentrations. And these are pretty tightly correlated. For vertebrate muscle and liver tissues, it was about 66%. And for vegetation and invertebrates, it was even higher. And so this really, again, shows that rare earth elements behave as a coherent group. And we wanted to know this because we wanted to know, do we need to test the individual elements, or can we sum the elements together, and that total or sum of all the elements together is a good proxy of how the individual elements will be behaving. And so this indicates that we can use the sum, the total rare earth elements, as a proxy here. And one thing that's interesting to note too on this graph is most of the points, the majority of them fall below this 1-to-1 slope line. And this really indicates that the light elements are more concentrated than the heavy elements. And in fact, when we looked, we found that on average, the composition of rare earth elements within organisms, and here it's all of the organisms mixed from all the ecosystems together, so it's over 300 data points. And we found that roughly 86% of the total rare earth elements was made up of these light elements. So then that was basically how the rare earth elements behave. And then we wanted to look more at more in detail at the bioaccumulation patterns. We wanted to know, do these elements bioaccumulate in northern ecosystems? And here this figure is showing our different taxonomic groups on here on the x-axis, and on the y-axis the total concentration. So as I was saying, we're not looking at the individual elements here, just the sum of all the elements together. And a first really interesting finding that we found, because there's not a lot of data out there on invertebrate concentrations for rare earth elements. And so what we found was that all of the invertebrates in freshwater and in the marine ecosystem had pretty significant concentrations of rare earth elements. So they have, here this is a nanomole per gram, so they're about 10 to 100 nanomole per gram of the total element concentrations. And this was high for all the invertebrates here. And this is, like I said, interesting because there's really not a lot of data out there on invertebrate concentrations. We also then compared it to the vegetation. So we looked at the concentrations in lichen and moss on the left and plants on the right. And these were also moderately high as well. It's known that lichen and moss do take up a lot of metals because they take up atmosphere. They tend to accumulate a lot of metals from the atmosphere, and this is probably what we're seeing here with the rare earth elements, as opposed to vascular plants that will uptake them via their root system, and potentially that's more difficult as rare earth elements can be quite insoluble. And then when we compared, so here we have our invertebrates and vegetation at sort of low trophic level species. And then when we compare them to all of the animals from the 3 different ecosystems, we found that across the board here, all of the muscle tissue concentrations were really low. Actually, a lot of them, especially the heavy elements, were below detection limits and quite variable. So this is really just saying that in these animal tissues from the 3 different ecosystems, we have really low concentrations, and you can really see the difference here between vegetation and invertebrates. Then what we did was we took these same muscle concentrations and we compared them to liver samples from the same animals, wanting to know if there was any sort of different distribution between the rare earth elements within the animals. And it was really interesting to find that the liver actually is significantly more concentrated in rare earth elements than the muscle. So really low concentrations in the muscle, but significantly higher concentrations in most of the species sampled here. And these liver concentrations were 400 to 400 times higher than what we found in the muscle. And some other studies have shown this, that whole fish samples will have higher concentrations, as rare earth elements also can accumulate in bone. So really emphasizing here that there is different distribution of these elements between muscle tissues and then internal organs like livers or kidneys, and also in the bones of these animals. We looked at a few other variables to see if there was anything else that was influencing the bioaccumulation, and I'm going to show you data for brook trout from the lakes and for ringed seal from the marine environment. These were the data sets for which we had the biggest sample size, essentially. And so here on the left, we have our brook trout from our lakes. And we tested size, so length and age, mass, and sex, and their relationship with the concentrations of rare earth elements. And we only did this for liver concentrations because the muscle concentrations were very low and quite variable. And in the fish, we found that there is a significant increase with age, so over time, for rare earth elements in the liver, and it's kind of a weak positive correlation. There was a lot of scatter here, but did increase by a little bit over 1 nanomole per gram per year in the fish livers. And what was significant, though, was the difference between the male fish and the female fish. So finding that male fish had, on average, 2.5 times higher concentrations in the liver than female fish. We hypothesized that this is due to things like metal depuration from the females when they produce their eggs. Could also be due to differences in foraging behavior, or even differences in how these elements are metabolized in the liver. It's to be investigated. When we looked at ringed seal, we also found an increase over time, or sort of with length. So as the seal's length increased here on the x-axis, we found increasing concentrations of rare earth elements. here, and we didn't find an effect of sex. Actually, it might be due to the fact that we had a low number of female seals sampled, or that there might not be an effect of sex for this species. And so this data together shows that these, although in animals we did find low concentrations of rare earth elements, over time they can increase. So over age and over length, we saw a significant increase in these species, and this really implies that the uptake of these elements is greater than their elimination over the lifespan of these species. And the final research goal that we had here was to investigate their biomagnification. So this is the idea that as we go up different trophic levels up the food web, we have more and more higher and higher concentrations of a contaminant. And so what I've done to show this is I've plotted here on the bottom axis the trophic level, and this was a proxy using the stable isotope ratio of nitrogen, so the delta 15N, and this really, as it increases, it shows you increasing trophic level. And we have our three systems here, and I've put in the lower level trophic level, so the invertebrates in the aquatic systems, and the plants in the terrestrial system. And then if I plot the mussel, we can see that with increasing trophic level, so as we go here to the left, we have decreasing concentrations of the rare earth elements. And this really confirms what we saw in the box plots earlier. And even when we compare these to the liver concentrations, we still see that typically the low-level trophic organisms have higher concentrations of rare earth elements. So I'm just going to summarize some of the results here. So we found that as they do in abiotic matrices, so as they do in soil and water, the rare earth elements tend to bioaccumulate as a coherent group. We did find some anomalies, but in general, there is a consistent pattern of bioaccumulation. We found that the highest concentrations were found at the base of the food web, and this is interesting if we're looking at bioindicators of rare earth element pollution. This indicates that we should focus on primary producers or primary consumers, as it seem to have the highest concentrations, and that muscle tissues of animals really do have quite low concentrations naturally of these elements. And also pointing out too that the different distribution between internal organs, so that livers have higher concentrations than muscle tissue as well, was quite an interesting finding that we are excited about. And finally, though they bioaccumulate and though we see trends over time in a few species, so that sort of across the board they can bioaccumulate and bioaccumulate over time based on their exposure, there seems to be limited evidence of biomagnification, so we don't see increasing concentrations as we move up the food web. And the study is, I think, a really interesting study because it adds sort of new information on their behavior and their bioaccumulation, and we don't know a lot about rare earth elements in general, and especially we don't know about anything about their behavior in northern environments. And I think it really shows overall that these elements bioaccumulate across a wide variety of different organisms. So from 3 different ecosystems, a wide variety of trophic levels, we do see bioaccumulation of these metals, and in sort of a consistent pattern across different ecosystems. So they sort of behave in a coherent and predictable way. And I'd like to emphasize that we do still need future research. We don't understand a lot about their bioavailability, about their uptake processes, and sort of all the cellular mechanisms of how they're toxic in the organisms. And it's really, I think, necessary to better understand rare earth elements if we are aiming to have an environmentally responsible exploitation of them in Canada. So I think that's about half an hour. I'd like to end now, and I'd really like to thank my collaborators, my co-authors, and my lab for their help on this project. And I'd really like to send out a special thanks to the hunters of Subarctic, because without them, this project really couldn't have been possible. And I'm happy to take any questions.
Thanks, Gwyneth.
Oh, I'm getting my
That's
guy.
really cool and really interesting. Wow. Thanks. Are there any questions from the audience, either live? We have Jérôme, and are there any people live on YouTube who want to ask any questions? I'm just going to wait a bit. I think you might want to.
What are you seeing this? Should I stop sharing and screen?
No? Yeah, that's good. Any questions for Gwyneth?
Can you hear me?
Yeah.
Okay. Hey, very good talk. I'm sorry I was late to start. I was stuck in another meeting, but very interesting. It's good to have good results like that. I had the questions linked to one of the lecture that Jules gave at the last SCL CCFFR. And I don't know if you were there, but Jules was explaining or showing that one new approach when we look at contaminations is to look at indicators that could be targeted indicators or non-targeted, more general indicators. And I was wondering if you are considering to look at potential indicators other than just measuring concentrations of particular contaminants. And I guess I'm asking this because do you have an idea of the potential cumulative effect of many other contaminants that are not necessarily the rare elements that could also interfere with some of the results that you have? I don't know if I'm clear enough. I'm sorry.
Yeah, no, so I wasn't at the meeting. I know that Mark, my supervisor, was, so
Yes.
I didn't see Jules' talk. It's an interesting question. I think I should emphasize that these concentrations we're measuring are far below the concentrations where you would expect to see any toxic effect.
Right.
Because these are pristine northern environments, part of the idea was to sort of analyze these rare earth elements before there was any mining activity. Yeah. And they really, even though I say, you know, invertebrates and plants were more concentrated, it's still quite low relatively to what we can see in China and some of the more polluted sites. So they are below concentrations where you would expect to see any toxic effect.
And there is no indication of cumulative effects, combined effects of one rare element with a contaminant that is there in much higher concentrations that will have a known toxicologic effect, or no?
No, I don't think so. And there's very little data on it as well, actually. Okay. So that's part of the problem, is the cellular mechanisms of how they're toxic, how these elements induce toxicity in different organisms is sort of poorly studied.
Okay. Mm-hmm.
So the answer is I don't know, but it's a very good question.
Yeah.
Gwyneth, you have a question on YouTube. So Brian is asking, for monitoring REEs in fresh water, the water itself, have you considered using DGTs, diffuse gradient films?
Yeah, that's a good question. I haven't. Sorry, go ahead.
Yeah, no. Brian suggests that they offer a time-integrated measure of accumulation, and probably it would be interesting to compare what you see in the biota with those DGTs.
Yeah, and I haven't seen any studies on DGT, so I think that would be a really good idea and sort of novel, because I think it would be some of the first data on rare earth elements. Typically, they weren't studied because they're quite hard to measure, but now I think, you know, technology, ICP-MS technology is really good enough to measure these low concentrations. They're really quite low in fresh waters, but I think, yes, DGT technology would be really interesting, especially this project. So the community I was referred to in George River, they want to set up a community-based monitoring program, and I think that would be really an appropriate technology to use in terms of collaborating between the local community and the scientific community, maybe able to put in you know, something that would monitor long-term, like you said, over time, the concentrations of these elements here.
Mm-hmm. I had a speciation question. Do you have a sense of what is actually the species of those rare element that is most abundant in water? Like, what would these? So I'm curious. Do you think it's mostly particulate?
Yes.
REEs that are just being filtered out of the water, or are they actually you know, internalized, or do they just accumulate as particles? I'm sure you've.
Yeah, I think that typically high DOC waters have much higher concentrations of rare earth elements. And so I think really that they are typically in sort of suspended matter in the water column.
So do you think they could be co-exported from the watershed, from the catchment to the lake via DOC? Would DOC be a good vector of those REs? Can they bind to the DOC?
Yeah, I think so. And there's a study I read showing that as you test water samples going north along a gradient of permafrost degradation, you get increasing concentrations of rare earth elements in the water column, probably related to input of organic carbon from degrading permafrost in the watershed, and with it the rare earth elements as well.
I see.
Mm-hmm.
So
I mean, they are considered like hard acids, so they typically bond to phosphate and carbonates as well. So that would be another source of their dissolved water concentrations.
There've been no evidence of toxicity to humans so far, are there?
Yeah, I have read some of the medical literature, and yes, they can be toxic to humans in really high concentrations. In China, there's a problem like it's not very good environmental protection in the factories that use rare earth elements, and the dust produced, the rare earth element dust, can be quite toxic. And there are some medical procedures that use rare earth elements for imaging. And depending, I know that some of them, if you have liver problems, you shouldn't be exposed to them as well. So there is some indication in humans of long-term exposure to rare earth elements can be toxic in very high concentration.
Are there any other questions for me? We're just waiting for the questions to come in. I know it's a little awkward to let you screen. I do have another one. If let's say a microbiologist would want to look at how they interact with bacteria, archaea, or even algae, would we need to work with the whole suite of those REs, or would there be a compound that is a representative type of the REs?
Yeah, that's a good question, and I think, depending on your research question, a lot of people that study toxicity just choose one element because they do they co-vary and behave in similar ways. And so, I think if you looked at one light and one heavy element, and it would probably be pretty representative of how the other light and heavy elements would behave. There's only 2, cerium and europium, that have different redox states, and we did notice some differences. Vascular plants did uptake cerium a lot more than the other elements, probably due to redox conditions.
Huh.
So there are some, and those probably wouldn't be good choices to choose, cerium or europium, because they behave a little bit differently than the other ones. But I think if you looked at the most abundant light and heavy element, then it would be representative of the other ones. Yeah.
All right.
Avoid having to test 15 different elements. So.
Well, I think Jerome had a really good question about this mixture, or the synergy of the effects. I'm curious to see how the field is advancing on that as we learn more about this cohort of elements that are supposedly behaving in similar ways, right? So, no, very good. Anything else? No? Well, that's great. Well, thanks, Gwyneth.
You're welcome. Thanks for being there.
Thank you very much for the good talk.
Yeah,
you're welcome.
Great. Let me just sign out to present this. So you'll be able to tune in back into look at Gwyneth's talk on YouTube as well. It's going to be archived on our website on the Society of Canadian Immunologists. And I'll be soon updating the list with new and exciting research talks. So don't hesitate to contact us if you want to participate. And I wish you a wonderful week. Bye, everyone.
Thank you, Alex. Bye-bye.
Thanks, Alex.